Okay, so we're going to talk about DNA and RNA as our fourth type of organic molecule. So we talked about carbohydrates. We talked about simple sugars, which are glucose, right? It's the most common simple sugar we think of. Fructose is another one. Then we talked about disaccharides. So the monosaccharides are glucose, fructose. And then the disaccharides, a popular one is sucrose, which is the sugar that we put in our foods. And then we talked about the polysaccharides, starch, glycogen, cellulose, and chitin. We talked about the lipids. We had phospholipids, steroids, right, and triglycerides. And then we talked about proteins, which are just proteins. All enzymes are proteins. That's a key concept you should know for our test. All enzymes are proteins. And we have many, many, many enzymes part of our metabolism. So we need these proteins for that enzyme function for metabolism. And we also need proteins for our structure, muscle, bone, ligaments, skin, connective tissues has a protein. So the nucleic acids then are the fourth group of major organic molecules. And again, what defines an organic molecule? One piece of information, one little sentence describes and defines something as being organic versus inorganic. What makes something organic? Maybe go to the front of the notes. What defines something as an organic molecule? Say it louder. Exactly. It contains carbon. That's the key piece that defines all organic molecules. So they're repeating units of carbon and hydrogen. That's the backbone, the basic structure is carbon and hydrogen. So chains of carbon, rings of carbon, but regardless, carbon and hydrogen with some oxygen added in, and then maybe some nitrogen and sulfur when you talk about DNA and RNA and proteins. So DNA and RNA are nucleic acids. And DNA is all about genetics. It has the blueprint for making proteins. And we said, proteins are hugely important for our structure and function. So the fact, whatever makes you, you is based on the recipe book for making you, which is found in DNA. So DNA contains genes, which are little recipes for different proteins. So if you look at a DNA molecule, it's a sequence of nitrogen bases that code for different amino acids, which make up a protein. So we're going to look at DNA structure in detail. We're going to build as a class a DNA molecule. We'll do that in lab this week, or next week, I should say, or I'll provide you something online to do at home. We'll see which route we're going to go with that. But there's two types then of nucleic acids. There's the DNA that is the blueprint for making proteins. And then there's RNA, which is a little bit different in its structure and function. And we'll talk about it's more of a supportive nucleic acid, that it helps with protein synthesis. So if DNA contains the recipe for protein synthesis, for making new proteins, DNA doesn't actually do anything, though. It kind of just sits there stuck in the nucleus. And one type of RNA, messenger RNA, copies that code from DNA, leaves the nucleus, goes out to the ribosomes in the cytoplasm, and assists with protein synthesis. So we're going to talk about that process a little more. But for right now, we're just going to describe nucleic acids. So DNA contains the blueprint for making proteins, and RNA helps with protein synthesis. So it helps carry the blueprint out to the ribosome for protein synthesis. So whenever we talk about where proteins are made, it's always on these small little organelles called ribosomes. So if we look at the structure of DNA, we can see it looks like a twisted ladder. You can see there's the orange side and then there's the blue side. Those are sugar and phosphates in a repeating pattern. So it has a sugar phosphate backbone. So if you look back at here at this, I'll zoom in a little bit better here. So this is one side of the DNA ladder. So if you untwist this coiled structure here, so if I untwist that into a flattened ladder shape, the sides of the ladder are made up of this sugar, which is called deoxyribose. That's what the green structure is. And then the little yellow circle represents a phosphate group. So that's the phosphate. So So it's a sugar phosphate, sugar phosphate, sugar phosphate backbone, and then bonded to the sugar. And if we're talking about deoxyribonucleic acid, the sugar is deoxyribose. And if we're talking about RNA or ribonucleic acid, we're talking about ribose as the sugar. So two different sugars depending on the nucleic acid. But bonded to that sugar is a nitrogen base or nitrogenous base. And there's 5 different nitrogen bases: cytosine, thymine, uracil, adenine, guanine. And based on their chemical structure, the more simplified chemical structure, a single ring, we call these the pyrimidines. So cytosine, thymine, uracil fall into the category of pyrimidines. And purines are adenine and guanine, and they're made up of the double ring structure. And what we find is that a purine always bonds to a complementary pyrimidine. So we find when we look at the DNA structure is that adenine always binds with thymine or uracil, depending if it's DNA or RNA. But if we're looking at DNA, adenine always pairs with thymine. And guanine always pairs with cytosine because of their chemical nature. They form hydrogen bonds holding themselves together to form the rungs of the DNA ladder. So when you look at DNA structure, the sides of the ladder are sugars and phosphates in a repeating pattern, and then the nitrogenous bases form the rungs of the ladder with the adenine of one side of the ladder bonded to the thymine of the other side of the ladder. And like I said, we're going to build a molecule together to really make this a hands-on type of learning experience. So here we can see in a more simplified way the sugar phosphate backbone or sides of the ladder. And then in the center, we have the nitrogenous bases. The A stands for adenine, the T stands for thymine, and then the C for cytosine, and the G for guanine. So that's what holds the two sides of the DNA ladder together, are the hydrogen bonds between those nitrogenous bases. And what do we know about hydrogen bonds in terms of strength? Are they strong bonds or weak bonds? They're weak. They're the weakest of bonds. So it's very easy to unzip this DNA ladder if we have the right enzyme to break that. And we'll talk about that when we get more into detail in protein synthesis. So DNAs and proteins tell us a lot about evolutionary relationships. Now that we can sequence the DNA of an organism, we can say that based on the DNA structure of this organism, it's very similar to the DNA structure of a related organism. Therefore, they must have evolved at a similar time or come from this particular ancestor. So it's really important to understand the DNA blueprint of different organisms because it tells us more about relationships evolutionarily than it would, say, looking at their structure or looking at their behavior or where they're found. Because animals transplant and are carried from different places by different factors, human factors, weather factors, right? Rain, wind, whatever. So we can't just look at because they're mostly here, they must be related. We have to look more, molecularly and look into the DNA. So the job of molecular biology is to look at those relationships based on DNA structure. So I want to show you a video here quickly of DNA structure.
Structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Williams and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases in length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of the strand are not identical. Note that on one end of each strand, the 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free. While at the other end of the same strand, the 5' hydroxyl group of deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of the strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
All right, what I want to do now is refer you to our DNA structure in Blackboard.